During our junior year, it was decided that we were going to do a research paper as our capstone project. With us both being interested in biology and genetics, we thought it would be interesting to get in touch with different doctors to see whether or not our hypothesis was correct, that the effects of trauma could be passed down through genetics. Throughout our year of research, we realized that our hypothesis was incorrect, and we were intrigued to find out how.
Known as the father of modern genetics, Gregor Mendel used his vast knowledge to study the inheritance of certain traits in pea plants. His observations led to the well-known conclusion that 23 chromosomes come from your father and 23 chromosomes come from your mother.
The functions of these 46 chromosomes were once unknown to the scientific world, similar to the manner in which the theory of epigenetics is now unknown to most of the scientific world. Epigenetics is the theory that environmental influences, including traumatic experiences and stress, can be passed down the genes of your children or your grandchildren.
Conrad Waddington, an English developmental biologist, suggested that something was working on top of the DNA sequence to modulate gene expression. Without the advanced technology of today, Waddington had a difficult time in the 1950s proving the theory of epigenetics. However, this was the beginning to a great scientific accomplishment.
Our initial thoughts before starting any research was that it was impossible for the experiences of parents to alter the genes of their children and even their grandchildren. To illustrate that epigenetics played a role in these studies, scientists would have to make sure that the family's medical history did not have any previous signs of illness that were not affiliated with genetics alone.
Take, for instance, genetic mutations. These mutations can affect the conclusions that researchers have come to regarding epigenetics because the changes in the genetic sequences of these children could thus be attributed to a mutation rather than solely epigenetics. Our main concern with the idea that mutations were causing these changes was the level of difficulty in proving it, seeing as organisms with larger genomes hide mutations without showing any signs of doing so.
A long-standing issue is whether the development of a human being depends on life experiences or already predisposed DNA. The question posed was if the way these children were raised would affect the studies of epigenetic inheritance. Individuals who have a severe form of depression show decreased glucocorticoid receptors, NR3C1, and increased HPA, also known as hypothalamic pituitary adrenal axis activity. The HPA axis, which produces glucocorticoids and releases them into the bloodstream, involves the hypothalamus and pituitary gland in the brain and the adrenal glands in the kidneys. The HPA is our central stress response system. These findings were why these researchers hypothesized that suicide victims would show decreased expression both of glucocorticoid receptor and glucocorticoid receptor 1F. A study showed that the expression of total glucocorticoid receptor mRNA was significantly reduced in suicide victims with a history of child abuse rather than those with no history of abuse.
A study done by Emory University showed that certain fears could be inherited through generations. Carrie Ressler, a neurobiologist and psychiatrist at Emory University in Atlanta, Georgia, became interested in epigenetic inheritance while working with people less fortunate within the inner cities.
Ressler decided to study laboratory mice that were trained to fear the smell of acetophenone. He decided that he would waft the scent throughout a small chamber while also giving small electric shocks to the male mice. These mice eventually learned to fear the scent because of the association that arose between the scent and pain. They even began to shudder in the presence of acetophenone, even without a shock. This reaction was passed down to future generations because despite the fact that they had never encountered acetophenone, they exhibited increased sensitivity when introduced to its smell. Ressler proposed that DNA methylation is the cause of this inherited effect. DNA methylation is a reversible modification to DNA that typically blocks transcription without altering its sequence. DNA methylation refers to the presence of methyl groups on the DNA bases, usually cytosine. It can also relate to the process of adding methyl groups to these bases. Rachel Yehuda, a researcher at Mount Sinai Hospital, set out to prove the theory of epigenetics. She studied 32 Jewish men and women who had been interned in a Nazi concentration camp, witnessed or experienced, or who had to hide during the Second World War. In every experiment, there is a control group, and this experiment was no exception, as it was Jewish men and women who were living outside of Europe during the war, and their children.
The team studied a region of the gene associated with the regulation of stress hormones that was affected by trauma. These researchers found epigenetic tags on the very same part of the gene in both the Holocaust survivors and their children, yet these epigenetic tags were not found in any of the Jewish men and women and their children from the control group. Epigenetic tags latch onto DNA and can either turn genes on or off. In addition to a previous study, Yehuda studied 38 women who were pregnant on 9/11 and had either been near or at the Twin Towers at the time of the terrorist attack. She studied women who had developed PTSD after the attacks and women who had not developed PTSD after the attacks.
For the future, these findings could possibly allow scientists to figure out how to pinpoint exactly where depression or traumatic effects are located so that they can bring them to a halt. Hopefully, this will allow future generations to decrease the number of suicides, depressed people, and traumatic sufferers. Specifically, when it comes to cancerous cells, it is epigenetics that most scientists look closely at. Disruption of the epigenome is a fundamental mechanism in cancer, and several epigenetic drugs have proved to prolong survival and to be less toxic than conventional chemotherapy treatments.